Good morning and good afternoon and good evening, everybody. My name is Christina Baxter, and I am going to be joined in just a second. You can see his camera coming up by Scott Hartley from ProEngine. And we're going to talk to you today about biological threats and where we are in terms of preparedness and detection capabilities and things of that nature. So I'm coming to you from my hotel room, so hopefully I'll have good connectivity throughout. But I'm here in Florida at the Defense Threat Reduction Agency conference. So really a perfect time to do a talk like this because there's so many new and emerging threats as well as technologies coming our way. And so we'd like to talk to you about a few of those today. So I'm going to turn it over to Scott to do introductions, and then we'll move from there.
Hi, my name is Scott Hartley. So kind of these webinars are really for educational purposes for it. So I'm not the one that's putting my life at risk for some of these events, but what we're really trying to do is to educate you about some of the threat environment and some solutions that are there that are made available. So we prefer that you ask questions on this. So please feel free there's a chat or a question button on your response. So feel free to ask those questions and we'll spend some time at the end of the webinar to try and answer those for you as well.
Perfect. I'm going to turn off the camera so we don't keep scaring people with our little happy faces, but you just have to hear us from this point forward. Just as a reminder before we go off screen is these events are recorded and that we will make them available to all of you afterwards that want to get more information. So in terms of today's talk, we're going to talk about the evolving threat and where we are. So if you've been in some of these talks with us in the past, we always start off where I'm going to go through where we see the threat today. And I'm going to pass it over to Scott and talk about some of the detection technologies, and then back to me on some of that operational environment type of information. So with that, I'm going to get started. If I can get the computer screen to go down one, that would be great. Okay. So what is bioterrorism? So first off, reality is bioterrorism is just the intentional release of these biological agents. It doesn't matter whether it's bacteria, viruses, fungus, any of these different things. In reality today, one of the most prolific things that we're seeing are the toxins, which as a chemist myself, and Scott is as well, we like the toxins because they're really chemicals. They're just derived from biological origin. And so it makes our life much easier. So we'll talk about the differences there. But reality is we are in a state of time where bioterrorism is quickly coming back into the forefront. A lot of that is due to technological advances, but a lot of that is also due to the environment around the world that we're working in. So whether it be a bioterrorism event that is an intentional release or just a natural dissemination of biological materials from other types of, or an accidental release, these are all things that unfortunately we have to deal with. So with that said, I wanted to go through a little bit about the impacts. So first off, generally we're looking at bioterrorism as something that has potential for high morbidity and mortality, mainly because oftentimes we don't know that this has happened until we start to see the signs and symptoms of the exposure, which could be days to weeks following an event. The impact that each of these things have is really dependent, not only the most important thing is how quickly do we identify the attack, but what was the agent, how much did they use, how did they disperse it, what was the weather when they released it, as well as the weather immediately following it, and then any of those pre-existing immunities of the exposed population. So if you were to have smallpox 30 years ago, we had immunity from a lot of vaccinated people. Today, many people aren't vaccinated against smallpox and brings up another vulnerability that we have. But reality is each of these things is going to be really important. Now, we used to say that we didn't have to worry about a lot of these threats because it was too hard for people to do. The impetus behind it has changed dramatically over the last few years. So just first, where is the threat coming from? Most of what we see is internet chatter in terms of the biological threat. A lot of that goes down to everything from what have we seen happen or misinformation campaigns like you see here on the screen right now. Each of these misinformation campaigns has been going on for several years now. And what it does is tells different people in different arenas that the United States used biological weapons against the people of Iraq and Afghanistan. And with those, it drops that level of should we or shouldn't we use these, because it's saying, you know what, they were already used. If we use them now, it's just retribution. And that's different. It's the dropping the barrier to using these types of materials. Now, once they came out with this misinformation campaign, they actually came out with a credible information campaign as well. And with that, it was a series of different videos that go step-by-step YouTube-style videos where they walk you through how to not only get a hold of the materials that you would need to make the threat, but also a step-by-step, how do you incubate it, how do you grow things. What really changed it now is they went into the dissemination and weaponization of the threats. And so, for instance, if you can't get your botulinum toxin to spread well through an aerosol, consider adding some soap, because that will make it break down into smaller droplets, which will spread more readily. That's the level of detail and why things have changed so much over the last couple of years. Now, each of the different videos walks through how you can take advantage of this, pretty much the same process, starting from acquiring the specimens. So I've highlighted in red the ones that I feel make the biggest difference today. So for instance, we can now buy commercial cultures for a lot of the biological threats. And a lot of this has to do with the expansion of the genomics work that's being done around the world. There are places where we can just go and buy commercial cultures today. A lot of places in the Far East where you can buy these. And most of these companies, while well-meaning, don't really understand the effects of selling some of these commercial cultures to the wrong people. If there's just pieces of the genetic code that can be altered. Now, getting materials from other scientists or from universities and hospitals where things may not be as secure as we would like them to be. Remember, if you're talking about something like a saxitoxin, a toxin from some of the shellfish, that you can have milligrams of it sitting on a shelf in a university laboratory without extra precautions, even though the toxic dose is in the nanograms. So we've got milligrams that are unprotected. And that's all based upon the requirements around the world. Now, what made it really interesting is the accessing technical information. Obviously, most technical journals and things are readily available online now. But what happened in a lot of these videos, if they've actually linked in the step-by-step process, and in some cases, they've gone through and videoed that step-by-step process. So when you start seeing the level of detail now, it takes away the fact that we used to think you had to have a microbiology degree to be able to do any of this. That's gone by the wayside. Accessing the equipment with the do-it-yourself biology programs and the fact that we have so many people at home doing home brewing, whether it be for wines or for beers and things, all of that equipment that is used for that is easily translated over to equipment used to amplify these types of threats. Culturing and propagating the agents, getting all of that material that you can get for growth medias and things are readily available today online. If we were just trying to follow different growth medias, we would have hundreds of people trying to track that. It's just not a feasible thing to do. On the weaponization, talking about how do you mill the material, what types of gliding agents or flowing agents do you add to keep this stuff aerosolized and airborne longer, all of that is now online. And then, of course, the dispersion, the use of UAVs and aerosol spraying devices, and what are the different ways that you can enhance the aerosol to make it stay more viable. Those things are all readily available, and each of these videos includes this step-by-step process, which makes it a very much more viable threat arena. Now, I wanted to go through a few of those different areas and just talk to you a little bit about that. One of the things that we're doing right now in the acquiring specimens is trying to get companies and even universities and others that are working with these types of threats to use what we call a Know Your Customer program for any of the dual use types of materials. And in that case, it's about understanding who you're selling things to, making sure that those people that you're selling to have a valid reason to have this material. So is it a commercial company that works in this area? Is it a university research program? Do they have grants from the federal governments that we all represent to make sure that we are getting the materials to the people who need it, but actually protecting it from going down other roads? Red flags that we always say to look for are people not giving you the appropriate amount of information, people that are trying to ship it to their home addresses. When you start looking up these addresses, making sure it's a viable company in our area where they would be working on these types of things. Making sure when you're selling or shipping material that you know where you're shipping to and who you're shipping to. And then make sure that we've got all of these materials flagged. It's almost like when we were first working on the drug community programs where we had people buying all the materials to make methamphetamine, but we put in place countermeasures that allowed us to say, if they buy this material and this material and this material together, flag it. And it's trying to get those types of things in place. Now, what I'm seeing a lot of, and spending a lot of time this week working on, is the digital biological data. The amount of data that's readily available that provides the DNA and RNA sequences of interest. And the fact that we now have whole genomic sequences as well as genetic sequencing equipment readily available that can go field forward and can provide us with answers, but also provide us with the opposite, an enhanced threat. So accessing the equipment, as I said, the amount of equipment that is out there today and the fermenters and those types of things, home breweries have made that so it's just quite easy to get a hold of. Centrifuges, flow filtration equipment, always been readily available. The freeze drying equipment that used to be harder to get is now being used at home for people to grow their own food and have it last longer. Sterilizers, obviously, with all types of materials that we have for sterilization. But then you get into the biological safety equipment as well. And there's all kinds of videos online on how to make your own flow glove boxes and laminar flow hoods. And how can you get yourself to create a safe environment for what you're trying to do, but also walking through and making these on the fly. Culturing and propagating the agents has become much easier, but it's all about that cell culture media, the nucleic acid assemblers and synthesizers, incubators, all of that stuff readily available to all of us out there. But what worries me the most is the genetic elements and that genetically modified organisms that are becoming readily available in the name of science. But in this case, we have to be much more conscious of the things that can go wrong here, because we are at a point in time where the science is exploding in this area, providing us with amazing opportunities for growth in the detection and in the understanding of the movement of threats. But it also puts us in an area where we have people that just don't understand the secondary effects. Now, reality is, we used to always say, people aren't going to be able to scale this up, so it's really not that big a threat. But today, we're really talking about targeted use. We're not talking about a state dispersion of a material. So reality is, if we're talking about a targeted or localized threat, when we can get 10,000 lethal doses of anthrax in 1 milliliter of material, that's a problem, because we can now get enough to create impact, especially as we look at these much smaller targeted arenas. And just kind of understanding that that large scale up that we were always looking at and saying, this is not going to be a problem because they're going to not have this capability. The capability is now there. It may not be the capability to take out entire cities, but taking out small focused populations is what they're really talking about today. Now, when you start looking about the production levels and start thinking about what would we need to do for like bacterial production, it's really not that hard when you start dividing it down step by step and start looking at the materials that they would need to get. So the hardest part is always going to be to isolate that source sample. And whether that be procuring it, stealing it, isolating it from other areas, that's the hardest part. Once you get from there and you start doing the culture, all of that is becoming incredibly easy today. Now, if we were talking a large scale dissemination, much harder because you've got to scale up everything. But at this lower level that we're really concerned about today, this is where we start to see things. And we can't control down to that culture level, trying to keep track of who has different agar plates and what type of growth media you're selling, just not something that we can keep track of. So those smaller cultures are the thing that I'm really worried about. In terms of virus, it's the same type of thing. It's that source material. All they need to do when they have that source material is to get that host to grow it. And then we can actually start the scale up process and incubation. Just seeing things like eggs that are tapped open on one side as a growth media, seeing blood agar, or just having blood areas. Some of the areas that we just look at is as simple as having chicken stock to grow in. So knowing that we have the ability out there in the step-by-step process, walking them through exactly what they need and exactly which type of growth media works best for each threat material. All of that is readily available. The one that I worry about the most today, obviously, is the toxins. The toxins, mainly because of availability. If you start thinking about the pictures I have here are just from ricin, but just walking through this process and knowing that, frankly, I'm in South Florida. I could walk outside and get enough material for ricin today. Understanding, thankfully, that most people in the US today that have made ricin have really gotten down to that most of them are just creating the pulp. So they're really just grinding up the material, not making it anything that is of high concern, but still problematic because of intent in the things that are there. But knowing that most of this is an extraction process and understanding that there's no real requirements to the storage of these toxins. Once they've been isolated, they just sit on the standard shelf. Not really a big thing. Whereas biological, I sometimes have to keep them heated. Some of, for the bacteria, for some of the viruses, we have to keep them cool. Things of different natures that when we're looking at it from an operational perspective, that will be drawbacks to them employing the material. The weaponization, the things that they've been looking at and discussing have all been about aerosolization devices. Now, if we go back to when we all probably first started studying this, we talked about people using planes to disperse this. Today, we're really focusing in on aerosolization devices as simple as UAVs. So pesticide delivery UAVs are talked about repeatedly, but reality is just the standard spraying and fogging systems that you use for the delivery of pesticides are being looked at daily for this type of threat. And this is where we actually saw the threat material that said, you know, hey, if you can't get your botulinum to spray well, go ahead and add some soap. Same type of thing we were seeing in terms of adding in dispersion materials from gliding agents and things of that nature. So I put this quote in here from Bob Kristovich from DITRA because he put this out the other day just before the conference. And he's saying, when we're preparing for this growing threat in the midst of a biotechnology revolution that is changing all of our thinking as to what is possible, this is what I'm trying to get at is we have now reached this biotechnology revolution, which has not only increased our preparedness potentially, but it has greatly increased our threat matrix as well. What used to be something that we considered too far off for our adversaries is now something that is readily available. And just by using slight changes in their thought process, we have now put ourselves at a disadvantage because we don't have the ways to readily, quickly detect and identify these threats. So I'm going to turn this over to Scott for a few moments, and then I'll come back on again in a bit.
Thanks, Christina. So I'm going to talk a little bit about the detection standpoint of some of these materials. So just to kind of set the ground a little bit, one of the challenges when it comes to biological materials versus chemical detection, for example, is that there's a lot of divergent considerations between detecting chemicals or chemical agents and biological agents. So one of the challenges is just the fact of their presence. So a lot of times in the case of chemical detection, you're detecting something that's normally not there. So when you see it, you're like, hey, this shouldn't be here. It's usually harmful, all that sort of stuff. In the case of biological threats is that they normally are there. And we as humans are extremely good disseminators of biological materials. We actually are good incubators of it, and we can spread them. And so from a standpoint of what we talked about before in the previous slide, between some of those materials and not needing a significant amount of material present, that creates a challenge for us, especially if we're in the middle of a large gathering. There's going to be more biological activity than if you took that same room and there's no one in it. And so just from a standpoint of detection, there's a lot more challenges with it. We do thankfully have technologies out there, and we're going to talk a little bit more in detail about those, especially Christina will, where if we can get a good sample of materials, we can use those to identify a lot of the materials for it. The challenge becomes in some of these cases, if you take, like, for example, the BioWatch program, sometimes it's knowing when to take a sample that becomes important. So our trigger mechanism and our trigger technologies start to become important for these. So we normally want to know when to take a sample because those tests take time. They also sometimes could identify something and it not necessarily be a threat profile. That's the other challenge with some of these things. So a lot of times what we do is we tend to look at the secondary effects and have to wait for that incubation period for some of those to occur. But if we can use, in case of, and we're going to talk a little bit more detail, a trigger in tandem with some of the identification technologies, you now are getting a hit on more than one solution at the same time, and that can help you out with that. So I'm going to talk a little bit more about some of this and how to use flame spectroscopy in particular, in tandem with that as part of your upfront, you're continually monitoring and flagging and using that as a trigger mechanism for identifying of some of these materials. So we're going to talk more in the next couple slides about some of that. So if you can go to the next slide, please. So again, around that same thing, so this is the way that one construct of way of looking at the problem. I'm not saying that this is the only way to look at it, but your ability to detect, to warn, and then to collect and identify becomes important. So you're only good as good as the sample that you have, and you need to know when to do that. So what you're really looking for is some sort of optimality that you can to then trigger those samples and then to take those technologies. The other challenge with some of your biological detection methods is just the time to detect versus in the case of a lot of on your chemical side, again, you can have some materials that can identify in a matter of seconds, or if you're doing a full spectrum and going through a GC mass spec, for example, which is even probably, which you can argue as probably the longest time frame in order to identify, you're talking minutes to tens of minutes of doing that, and that's not even where most of the biologicals can even identify something within that standpoint. So it is a very different means of defining the technology for it, and kind of you need solutions along this whole perimeter in order to create a holistic response to biological detection. And just on the chemical side, the same analogy is you want multiple detectors working together to kind of correlate and to tell you the same thing. You just don't want to rely on one technology or one detector in particular to do that. You want to get the same answer from multiple. The same thing comes as part of a holistic biological response, is that you want to get similar information from multiple detectors working together in tandem in order to help better identify and create a more holistic response to identifying and warning people in regards to some of these threats. So if you go to the next slide, so I won't go into a great detail about it. Christina hit on some of this, but we did spend our last webinar, we spent basically an hour talking about this, but it is important from the standpoint of a couple of things. One is most of your biological materials will be released as aerosols in order to be effective. And just because they're in an aerosol form, there's more ideal aerosol forms that can maximize their detection. So Christina was just mentioning some of those aspects of it. So they've learned some terrorist things is that not only just releasing it, but releasing them within your kind of your sweet spot that'll have the most effect on the human body to get into our respiratory systems, either in our upper or lower respiratory systems, is very much dependent upon the type of particle, the size of the particle, and how well you disseminate it. Plus, when you're wanting to disseminate these in a quote unquote weaponization means, you want them to stay in the air as long as possible. You don't want them to drop out of the airstream, right? So there's certain things that you can do, from controlling particle size, from adding in surfactants and other sort of things, plus a means of how you actually aerosolize them in the first place, that will make it a more effective aerosol. But the important thing is that these will be released as aerosols most likely. They will tend to be more localized. So it's not like you're going to have, unless you're dealing with a nation state actor that can make these on large scale. In many cases, it'll be a relatively a start and a stop with not a very long duration of those, where you only have a certain amount of material, but you tend to want to use them as targeted attacks, or in certain areas where there'll be aerosol releases over certain time periods, but it won't be over a whole city. It'll be over a smaller area, and that'll come and go. So your ability to identify and know when to take these samples is important, because there's only going to be a limited amount of time where you're going to have those materials at high enough concentrations to be able to identify them, and you wanted to take a sample and capture at those time periods. So that's important. So both particle size as well as the quality of the dissemination, and being able to identify in this kind of sweet spot where you're having that impact is important from a biological detection standpoint. If you want to go to the next slide. So what I'm going to show you here is just an animation of what occurs in flame spectroscopy. Some of you have probably seen this. Some of you may not have. But what you're seeing here is what is going inside of what occurs inside of a flame spectroscopy machine. So what you're seeing is an incoming airstream. So there can be aerosols in there. There's no filter ahead of there. Usually, because you are in a flame, and there you can and potentially have flammable environments, there's usually a pre-burner, which is what you see there. And then this is actual flame. So what the flame simply does is an energy source. That energy source is designed to excite the incoming airstream. And when you excite the airstream, you basically bring it into you excite the atoms, and as they release and come out of that stream, they release photons, which we can collect. And each element and material has a unique response specific to that element and that element only. So what we're doing is capturing that information through a series of lenses onto a CMOS sensor. The CMOS sensor in particular has been an advancement that has really helped on a flame spectroscopy standpoint, because it allows us to capture the spectral information, and then we can run it into through a processor to signal process, where we're looking for the presence of that specific fingerprint associated with those elements, and those elements only for it. So this is what is kind of occurring inside of flame spectroscopy machines with it. There's multiple people that you can get these things from. Kind of our Pro Engine's claim to fame is our ability to detect multiple materials simultaneously, and to miniaturize and militarize it from the standpoint of militarization, from the standpoint of making something small that you can hold in your hand. Now, most of you are probably familiar with the ability to detect, use this technology for chemicals, but in actuality, you can actually use it for biological materials as well. So in the case of biologicals, we're still looking for the elemental analysis, but we're looking for 3 channels of information, and that's sodium, potassium, and calcium. So I'll talk a little bit of why those elements are particular importance to us, and why we tend to focus in on them. So if we remember back from our COVID days, some of you probably don't want to remember those, but there was a lot of talk about RNA vaccines and other things like that. So RNA is used a lot of times. If something is living, it'll have RNA presence in there. And if you look at some of the core materials needed for RNA for synthesis and growth and maintaining of of biological materials, because Christina talked about that as well, is that not only you have to generate some of these materials, but you have to have those materials maintained until you want to release them. So a lot of times they're using materials which contain sodium, potassium, calcium, because those materials need that in order for the biological materials need to continue to run. What you see to the right is a test chamber that we have. So we've released hundreds of different types of materials and ton tests on there, where we're looking, where we have our detector actually inside that chamber, and we're releasing various surrogates, so your spores or bacteria or viruses, and we're looking for the specific response associated with those. So each, similar to kind of on the chemical side, there's specific fingerprints or ratios between these materials that delineate between spores, bacteria, viruses. So we have all of that information, and we've collected that into there, so we can use that to identify between living and non-living materials, and we can also surrogate between, it's not impacted on other materials such as fluorescing, so we aren't determining on fluorescence, so there's things that fluoresce that aren't biologicals, for example. So we're able to more precisely identify between living and non-living materials and provide an alarm as far as that goes in the process. So that's kind of the juxtaposition of the process. So the one is we're taking the airstream, we're applying it to the process, and then we're collecting information on sodium, potassium, calcium, and then we're running it through and looking for the specific ratios. So it's not just the presence of those materials, it's presence of certain ratios of those materials in there allow us to identify and biologically determine with that. So that was our Gen 1 detector system that we had. And we know for, when I say Gen 1, basically we knew those fingerprint responses that were present in those responses. And so we could use that to determine it. The challenge becomes, if we go to the next slide, is the environment that you're in. So one of an additional challenge of biological detection is the amount of biological materials that are just present normally in an environment are very much dependent upon where you're at. So if you're in a city versus the country, the amount of baseline biological materials will be different. The climate that you're in. So if you're in a hot, humid climate, you're going to tend to have a lot more materials present than you will if you're in a cold climate such as Alaska in the middle of the winter, for example. So versus Florida in the middle of summer, just your baseline amount of materials were associated with that. Plus, you also could have near the ocean, and so you obviously see one of the materials we're looking at is sodium. So sodium likes to be in salt water. So one of the challenges that we were facing with that is that we needed to really have a better means of understanding the fundamental baseline and then using trigger mechanisms to do that. So those ratios of sodium, potassium, calcium and the response to the things were well understood. What we had to do is a better job of understanding the biological baseline and then being able to take consideration from that. So if you go to the next slide, what we're now doing is setting up and first understanding the signal background that's present, or the amount of baseline biological materials that are present in the material. An additional challenge of these is that you also, in a military standpoint, or even just any sort of use case scenario, these materials are designed to be attached on vehicles or other things like that. So if you think about one scenario is, let's say you're on a mountain in the middle of nowhere in a military base, and you're starting out your day, and then you're going on into a city to then monitor a biological detector. Your baseline function is going to be drastically different from when you're starting out in the beginning of the day to when you're entering in to the urban environment where you're doing your monitoring. So the system must be able to adjust appropriately to that environment. So what we're really doing and what we've added in is machine learning capability into the algorithm that we've developed that really allows us to adjust to the continually adjust to the baseline, as well as using statistical processes to make sure that when we are seeing a triggered event, that there's high probability of triggering an event from non-natural means, because the biological baseline, one of the additional challenges with nature is it's not something that's just a flat line, as you can kind of see there. There's going to be some ebbs and flows associated with that, which are just normally going to occur. So the advantage of some of the new machine learning technologies that are out there and the processing capability allows us to adjust for that appropriately. So that's really what we've implemented into our Gen 2 system that's now taking into consideration that and allows us to learn that background. So then we can delineate between the environment and the actual event environment that we're wanting to determine, and then we can use that to properly alarm and flag off of a device with it and flag off of it. So if you go to the next slide, this will kind of show, no expense was spared in our PowerPoint experiences here, so I'm just saying that a little facetiously, but what we're kind of seeing here is you have a detector in an urban environment, and it's sitting there, and if you you can click it and it's going to start a little animation. So the detector's up and running, and then there's a release of a biological cloud that's going over there. It's waiting to hit the detector, and then you're getting a response beyond that of the environment, and then you're using that to alarm and to flag, and then it'll go back out of the steady state. So in this case, what you're wanting to do is obviously take that sample and that trigger point where the anomaly occurred with it. So in the case of a lot of detection methods, what you're really trying to do is make sure you don't alarm when you aren't supposed to, but make sure you are alarming when you are supposed to. And there's always challenges with that. And so the advantage of the new machine learning background is allows us to take in consideration those changes in environments and still use the understanding of the proven ratios that we've determined are functions of those different biological materials, and then use that to trigger. And then you can create samples and use that for further identification of and validation of those threats. So you go to the next slide with that, and this is kind of, we'll talk a little bit more about that, is kind of where these systems fit into place is on the quicker detect to warn to trigger some of those other materials, where we can trigger very fast, let you know some sort of anomalies there. And then we can take those samples, and we're going to talk some more about, once you have a sample, some of those things that you can use to determine and to bring, use that to identify and to validate the response. So you're getting, when you get hits on more than one system and they correlate, then you have much more confidence in the result versus if I took a random sample and I see something that can be naturally occurring, for example, is that helpful information? Obviously, yes, but is that indicative of some sort of purposeful release or something I need to really worry about? I don't always know that. Versus if you have some of these things where you're saying, hey, I saw a pretty high level of biological activity that's beyond normal, and then all of a sudden I get hit for something like, for example, say smallpox, you have much higher confidence in the ability to say, okay, I need to probably really start to pay attention of this, because you're having two things or even three things that are saying the same answer to you, plus the higher level of biological activity that's beyond what should be normally seen is also indicative of something that I should need to be paying very high attention to and relative to threats environments that are being seen. So you go to the next slide. Pleased with that. So this is just to highlight a little bit. So this is the Gen 2. This is a VB, what we call VB system. And we also have what's called an AP4B. So we love the acronyms with it. So the AP4B is a standalone biological detector that has this built into it. We also have AP4C VB, which is a combined chemical and biological. So if you're doing the biological process, you could, in theory, just add the same chemicals that we're looking for. So adding in sodium, potassium, phosphorus, hydrogen, nitrogen bonds, arsenic, and sulfur, which are your main chemical warfare agents, as well as hitting on a large majority of your toxic industrial chemicals from an elemental analysis, being able to identify those simultaneously. So you can have these built into one package where you're having a simultaneous chem and biological, or you can have a standalone biological if you want into there with it. An additional aspect too, we have tested it in, we tested it outside in the middle of pollen season in the spring. With that, we tested in marine environments near diesel fumes and other things like that. Again, because there can be fluorescence with some of those materials, which can trigger some other systems. this is not able to be done. The baseline is set for 100 ACPLA. So ACPLA is the agents containing per liter of air associated with it. It's kind of a measure of a uniform kind of way to look at it as a kind of like a part per billion or part per million. But in the case of biologicals, it's the amount of biologicals in a liter of air, and the amount of threshold. The lower the number is, the lower the threshold that can be deciphered in there. We are adding shortly, that's going to come out, is the ability to tailor that trigger level based on customer desired levels, so you can lower that down to lower, lesser amounts, and then also trigger that faster if you need to as well, associated with that. If you want to go to the next slide with there, I'm just going to show real quickly how this can be used. So some people are like, okay, I have this detector, how do I use this or tie it in or other things like that? So just to let you know, this was shown at a recent trade show. It's a picture there. In the center, you see a display, and that display is showing data from multiple chemical detectors. So some of these are not pro-engine detectors, they're single gas detectors, as well as biosampler systems. So both wet and dry biosamplers that can be triggered from the detectors. So on the right-hand side, you have the AP4CV, which is the chemical detector version of the V, and then on the left-hand side, you have the biological. They're all feeding into the channels and able to be monitored and trigger and show alarms associated with all of those. And then you can add in radio stations, GPS, other sort of things in there, as well as bring in other sensors from other locations and roll them under it with it. So it's just a display unit in the center that ties in all of these materials for there. So it does also allow for functional autonomy, where customers that don't really want to publicize what they're doing, but they want to put in a couple different detectors in a couple different locations. They don't want us to know what that is. That's fine. They can use that to set up their systems however they want, and then use it to monitor with it. If you just go to the next slide, in addition to there, this is just showing you, like, for example, a screen capture on a simultaneous release on something that contains both phosphorus and sulfur. So additional aspect of or benefit of flame spectroscopy processes is that you can have two distinct materials present in airstream simultaneously, and we can see those. So you're seeing two releases of those. So there's a release, and then that goes away, and then a second release associated with that. And then we also have the biological materials we can add in there as well into the system. So just showing you kind of a hypothetical approach on some of those that can be done. And we are capturing data on the chemical side in either micrograms per meter cubed or milligram per meter cubed, concentration of each of those elements. And then on the biological, it's more related to an ACPLA trigger level associated with some of those materials. It's set for 100 as part of the baseline on that. So I'll kind of turn it over. That's kind of a baseline of how to do that. Christina's going to hit on some of the more of the operational aspects, as well as some of the additional technologies for identification that could be used in tandem with this as part of a holistic response.
Okay, so when I start, everybody knows, like, if that been to some of our talks knows I love my red, yellow, greens here, but when I start looking at my biological aerosol detection toolbox, I have to remember that there's very few technologies out there today for capturing those aerosols in the real-time world that we work in. So in terms of the Q systems, the things that we're focusing on operationally is very much using something like this to cue an alarm and tell us when should we take a sample. Now, it's one thing if you're on a call and it's a white powder call or something of that nature, where you have a sample presented to you, or you have a very definitive sample that you can take a aliquot of. That's one approach, and we've done webinars on that approach. But what we're talking about here is having an environment where something has distinctly changed, in understanding that change quickly enough to be able to get a sample so that we can get ahead of the infectious dose starting to get into people and spread to others. And so in that case, we need a Q system. So that's what we've been talking about, are these systems that we can use, that we can Q at a level that is well below infectious dose. and then use that to cue something else to collect sample, whether that be a filter device, however you want to do that. Those are system agnostic. And then the identify devices. And in that case, I'm going to talk a little bit more about that one, because those, while it's also system agnostic from a ProEngine perspective, there's different things that we can and can't use in field operations, and different reasons why some work better than others. So when we start looking at what we use in the field for detection technologies, usually divide them out to a couple of different things. We have ATP testing, which all that tells us is, was that material from biological origin? Doesn't tell us anything about viability or whether it's a threat material or not. It just says, hey, this is biological in nature. The protein tests that a lot of us use, and that just tells us, does the material contain a protein? That doesn't tell us about all the different materials of interest, because many of the threat materials don't contain proteins. When you start talking about viruses and other things, or when you start talking about some things that are spore forming that you may not be able to detect as easily. So protein tests and ATP tests, we usually use for determining whether we have decontaminated an area. Have we cleaned that surface enough to be able to tell us something about reopening that space, even if we had to send a sample to a lab, and that lab may not get us answers for days. This allows us to remove the material of interest, disinfect or decontaminate the area of interest, and then do a couple of different quick tests to say, you know, there's nothing left of biological origin on this surface, and therefore we can open up that space. Now, when we get to identification, most of us in the field use either immunoassays or polymerase chain reaction types of materials. In this case, both of these require us to know some of the threats, or at least focus in on threats of interest, of high material interest. So most immunoassays, we have generally about 8 different threat materials we're looking for. And then the PCR, depending upon which one you have, about 27 to 30 different threat materials of interest. Now, those ones are more when we're trying to identify the threat. Now, we have to be really careful, because a lot of these threats we can't identify at levels that are operationally relevant, and we'll get into that a little bit more. Now, I wanted to show you a couple things when I was playing around. So obviously this is in the hood, but I was playing around with just some castor beans to say, can I detect the protein in the castor beans just using my traditional AP4C? So what this video is going to show you is, I did not aerosolize or any of that. What I did here was just crushed castor beans. And what I was trying to figure out is, can I see that disulfide bond? And so I'm going to just play you this real quickly. And you can see this is just a little bit of ricin. I'm taking the AP4C with the S4PF sampler. And all I'm doing right now is heating that up. So we're thermally desorbing some of that sample off into the AP4C using what we traditionally call the crack pipe. But it's a thermal desorption interface for the liquids and solids into the AP4C. You can see that sulfur bond coming up there. And that's just like doing a protein test, but using your AP4C to do that instead. So you don't have to buy a bunch of protein tests. It shows us the sulfur bond pretty quickly. And most of our polypeptide chains are linked by a disulfide bond. The other way we can do that is also looking at something like the hydrogen-nitrogen bonds. So in this case, that's what I was using for things like nicotine. When I was doing these videos, I did not have any high purity nicotine extracted at home, and it was not a safe extraction for me to do there. So in the hood, I just pulled out some caffeine to show you this example. But in this case, we're just going to show you caffeine and getting that nitrogen bond. So this is the HN bond. This will be similar to what I do when I'm doing the detection for the nicotine samples. And nicotine, as you know, has become one of the more prolific toxins in recent years. And all I'm going to do here is show you how that nitrogen bond shows up. If you watch in the second channel from the top, we'll heat up that sample, and you do have to heat it up on the S4PF to get to the higher level. And now you can see that nitrogen showing up. That's exactly how we see it on the nicotine extractions. Now, as I said, we have to be really careful about limits of detection for a lot of these. And so in my red, yellow, green matrix here, green means that we are measuring the material at the operationally relevant level. What that means is the limit of detection of the capability is telling us a level below which is the lethal dose. Okay, so we're actually able to tell it before somebody's going to have an infectious or lethal dose. Now, when I have it yellow, it means I can detect it, but it may not be as operationally relevant as I would like. This is very common in the biological world, that many of the things we can detect, it's still not able to get down to the level at which it's infectious, which means it's very difficult for us to clear a scene where we're downrange and say, if we think it is a potential threat, we must first package sample for the laboratory and send it that way. Then, if we're doing some of these field techniques, that is something that we're doing as a background quick field screening to say, is this space something that we're really concerned about, and how do we clean it to a point where we can then put people back in the space? Because as you can imagine, if we just use this and say, for example, this is on Capitol Hill and there is voting going on, but somebody just received a material, I need to be able to screen that material and say, you know what, we're going to send it off to be detected because they are a high profile person, which means that increases the threat level. It's a package that has interesting writing on it, some of that nature. We're definitely sending it to the lab. But now what I need to do is say, how can I get this space back open because we have votes coming up and we can't just shut down Capitol Hill. Doesn't work that way in the real world. So these are things that we use as quick references to say, have we gotten it below certain levels? The bad thing is we very rarely can tell if we've gotten it below infectious dose. So the different technologies that we talk about, our handheld immunoassays are very common amongst a lot of us. So there's a bunch of different types. In most cases, with the exception of some of the toxins, those cannot measure down to the level of infectivity. And so we give them a yellow, even though there's something that we're widely using. Now, when we get to some of the PCR techniques like that from the BioFire or from the biomeme companies, those technologies provide us a broad spectrum of threats that we're able to detect at operationally relevant levels and in a time period which ranges from, we'll say, 35 to 65, 70 minutes. That provides us, even though it takes a lot longer than most detection schemes that we use in the field, the capability to identify some of these things at operationally relevant levels. Now, does this take replace our detection in the laboratory? Absolutely not. Every time we have something that we feel is a biological threat, we need to make sure that the pristine sample, actually any type of threat really, if we think it is an offensive sample of some sort, whether it be chemical, biological, radiological, the first thing we must do is capture a pristine sample that can go to the laboratory, not only for the evidence perspective, but also for the ability to test it in an environment where everything is controlled. So all of these things are going to be considered presumptive in the field. Definitive is always going to go back to the laboratory. We have a very pristine environment and we can control every little thing we do. The PCR technology that we use today are the closest we have to giving a really definitive answer in the field versus a presumptive answer. And the reason behind that is, and I'll show you with the biomeme, is they actually put standards in place that allow us to go through and make sure the system is working properly for every sample. And that's the difference that we have today with some of these new technologies. Now, if you're talking about field immunoassay techniques, this is one of the ones that we've been looking at a lot more recently because it allows us to do a multiplex type of approach. And it's called the SAMI, so the Surface Sampling and Multiplexing Indicator. As you can see on the sample here, you twist out this top part, and that is your sampling wand. And you have a sponge here that's wetted. And so that's assuming you have material on a surface. You go, you get that sponge, you take your sample, you insert it back in, okay, until it clicks, and then invert it one time just to get that mixing going. Then you rotate the plunger over one, and that actually allows it to push your sample down into your multiplexed immunoassay test strips, where you pick which strips you want based upon availability. We use this for bio, we use it for some of the drug immunoassays. We also use it for some of the explosive immunoassays, which is kind of a really cool technique to be able to use this same approach for a lot of your different things. What I like about this technique as well is there's actually one more step you can bring it all the way over to the left-hand side. You can see where you can bring your sample all the way over and then down. What's at that point is it has saved a second sample. So what's doing is when you took your sample and you inverted it and then you pushed that plunger, it actually went through and gave you two different sample aliquots. One sample aliquot is going to go through and run your immunoassays. The second one goes into an isolated chamber within the sampling device, and that one is meant to go back to the laboratory. Now, that's a fantastic way to do it because you actually don't ever access that sample. Once you've taken it, it goes in, it gets into its buffer solution, and it stays right there. You send the entire device with your immunoassay results to the laboratory, and now they can extract that sample and have an exact duplicate of the sample that you took in the field. Now, what I've been doing a lot lately is playing around with this biomeme system, the biomeme Franklin. And what I was telling you before about the positive controls, you can see right here, there's a first positive control telling us that the actual system matrix is working properly, which is the system platform. And then the other one that's saying that the whole flow is working properly, that we actually had a sample that was in there and it made it through the entire system to show us, hey, this system's working. So you actually have your positive controls that you would do in a laboratory, but you have them out in the field, which is very cool now. Doesn't take away the need for doing things in the field, in the lab, but it does allow you this capability. This entire system is about 4 pounds. What I really like is the fact that I can now take a sample that I took in the field, and I think it's a 200 microliter aliquot of that sample that I've put into a buffer solution or whatever, if I've collected it as an aerosol device onto a filter, and then washed it into a surfactant of some sort, not a surfactant, sorry, into a buffer solution. Then I take that 200 microliter aliquot and I put it into the top of this sample device. The sample device then does everything else for me. So I'm no longer needing to do any of that front prep for PCR samples. All I'm doing is taking my sample, putting it in buffer, and putting it onto this device, and it does everything else for me. So with this case, you can see here that I had a ricin sample, and all 3 of my ricin indicators were positive, as well as my positive controls, and we now knew that things were working. What I was showing you here on the second one in the middle is one where my ricin was positive, but I also had a positive on this Brucella thing. And the reason I show you this one is this is actually skin contamination of the sampling. So in this case, we weren't wearing gloves when we were playing around, and you can actually see that we got a skin contamination in there. Now, it doesn't come up as a positive for Brucella, because for Brucella, you would need to have both of these, the one that would be infectious as well as the one that we're seeing just in skin contamination. And I know the company's going to remove that one, but I think that's kind of cool in terms of telling us, hey, you know what, be more careful. I also have this one here for botulinum that I showed you. And in this case, this is a botulinum vaccine that I used as a sample, and it allowed me to get a positive hit for botulinum. Now, there are different techniques you can use with these and get samples that are, you can run that will have fake answers and these things. I just like to play around with some of the real stuff and make sure that I do get the answers I'm looking for. And so I've had a chance to play with this one and had some great results. I've also played around with the BioFire system, mainly the benchtop system, because they have a lot more capability in their benchtop system for the film array, and very similar types of results. So when I'm talking about a benchtop, the film array does a lot of that, whereas this one is the newest of the handheld versions, 4 pounds, field ready. Now, because I'm at the DITRA conference, one of the things that I've been looking at is where are we going for this? Now, again, like I said, when we were back at this slide, in this case, we have to know the threat material because they're building us technologies to scan for those specific threats. But what if I don't know the threat? What if it's something new or something that's been modified? And that puts us into that genomics. And I didn't put any slides in here on this because it's still a bit out from what we're doing in the field. But the future of this is that we'll be doing whole genomics, where we'll be taking a sample in the field and actually getting the whole genome of it and then comparing it back to known libraries or looking for different infectious clips of that type of thing. Really crazy, the level of data that they're doing. But with all of the new artificial intelligence and data platforms that we have today, it's really exploded this whole area. The thing that I saw for PCR itself is, in terms of this biomeme, it takes me about 60 minutes to run the full sample. But where they're going is miniaturizing this even further. But their new system that they're working on is looking like it's going to be about a 10-minute runtime to be able to uniquely identify the threats that are within their matrix. And they're working on, right now, they have 27 threats. I believe they're working on a total of about 45 threats. That gives us such a capability. Obviously, the things at the DITRA conference are a couple of years away from fruition. So this is what I'm focusing on now. And this is the new technologies, is that horizon scanning of where we're heading. With that, for those of you who don't use PCR, the reason that we use this is that it allows us to amplify the results. So while we may only have a couple of different threat materials available in the real world, what we're capturing in that aerosol, we're able to continuously split it and replicate it over and over again until we get up to that 1 million target range that we need to get unique identification. And that 20 cycles or so is giving us 1 million targets. And so that's generally around that probably 35, 40-minute mark. We're going out to around 60 minutes to give us pretty much about 100 million targets. And the one thing to remember here with PCR is that PCR does not tell you whether it is still a viable threat. Meaning, the first step in PCR takes the threat and destroys it. Okay, what we're doing is denaturing that DNA and splitting it apart. And so while PCR will tell you in many cases what the threat is, it will not tell you whether that threat was deployed as a viable material or a non-viable material. So still not everything that we need in the field, but we're getting so much closer. So I want to end with just reminding you that there are many signs of a biological event. Unfortunately, we still have a lot to do. In most cases, we identify a biological event through epidemiology. It's generally a retrospective look back at something that happened in the past. It is not something where we can generally go and just take a quick sample like we do with chem and be able to do things. In this case, we're looking at epidemiology results. We're looking at casualties in a short time, increasing those number of casualties. Fatalities that don't make sense, okay? So when you start looking at all of that, that's where we are today. If we try instead to incorporate some of these trigger and cue technologies, that would allow us to get that positive result much earlier, because we can look at the background environment and say, here's our traditional background, but something interesting just occurred. We don't know what that is, but let's go ahead and take a sample now and bring it back to a lab or run it on one of these field forward instruments and find out what we have. And that's what we're trying to do, is get ourselves in front of the biological event at that point of infection before the incubation period has gone through and unfortunately spread a lot of that material before anybody was even symptomatic. So when you look at that pre-symptomatic but infectious period, that's what we need to get ahead of. And today, the only way we get ahead of that is by using trigger and cue systems to force us or let us know when to take a sample, because it's just not cost effective to continuously sample all the time. And that would just require for us to take things back to the lab over and over and over again. So with that, I'm going to turn it back over to Scott for any closing remarks, and then we'll look and see if we have any questions. But this is our contact information for both of us. And please feel free to reach out anytime. As we said in the very beginning, we did record this webinar. And so we will, through the ProEngine Academy team, will make it available to all of you that registered and participated in the webinar for reviewing and things later. So Scott, do you have any comments before we go into any questions?
Just one additional comment was just around ACPLA, which tends to be the measurement of biological materials present in a stream of air. I think similar to on the chemical side, there is a dependency of the threat environment based on the material you're dealing with. So for an analogy of that would be, so if I'm dealing with 2 parts per million of sarin versus VX, obviously they're both harmful to us and both of concern, but the ability to be to kill multiple people is higher with one than the other just because of the toxicity of the material. Some of that same analogy is present in the biological. So we do use ACPLA as kind of a baseline measurement, but there are differences into the threat profile based on what those concentration levels are and what the operational significance of those measurements. So do you want to be at 10 or 100 or 1,000? ACPLA will vary as well. So it is, sometimes it's a little bit hard concept to understand, but it is like a baseline that people are using a lot of times from a test methodology that's utilized to baseline some of that. So I just wanted to add that part to it.
Okay, we do have a couple of questions here. So the first one was just a comment that said similar companies outreach to explosive or drug precursors. Absolutely. Another one from Chris, do all typical biowarfare agents possess some ratio of potassium, calcium, and sodium?
Yes, that is yes, they do.
Yeah. I was like, I'm not sure how to answer that, but yes. Do we want to tell everybody what that is? Probably not.
No.
But yes, they do definitely have potassium, calcium, and sodium. Now, with that said, when you get to things like some of the toxins that are chemicals that have been derived from a biological, if there is no residue of the biological origin, let's say for ricin, the seed or the mash that it came from, then you would not see that. But same thing as you would not see in PCR or any of that, because you wouldn't have the biological origin material there.
Yeah, and
slide 38.
Yeah, and to add to that a little bit, I mean, we kind of treat, but biological toxins tend to fall under the quote unquote biological detection, but for all practical purposes, they're chemicals and they're more a chemical threat with it. But I would say one advantage potentially is if you can simultaneously look at both, and that's preferred just because those can be present. In some ways, as we talked about, the toxins are a little bit easier to get a hold of, probably a little bit easier to disseminate to a certain degree, but they do behave and act on the bodies not similar to how biological materials would do. They present themselves more as chemicals, but the ability to detect both is important because they need to be there as part of a holistic response.
Okay, one of the questions on the one where it was the floating cloud of biological agents coming across.
Yes.
On the background signal appeared to be higher than the original signal. How do you distinguish them?
So first example, I would say, we were just using a Excel PowerPoint and the limitations of that to kind of demonstrate it. But so to go back to what we're doing, so we're using machine learning technologies with that. So without getting into all the specific details, which tends to be a little bit of priority and not something I really want to discuss on an open line per se, but we are looking for trends and and taking consideration the overall thing. So not just one data point will cause it to flag, but you're looking for an overall response that will change with maybe.
Oh, I kind of lost you there for a minute, Scott. Oh, did we lose Scott? Okay, I'll go on to the next one here for a minute. Scott, are you back? Okay, I don't, we've lost your signal, it sounds like. On slide 23, slide 38, are you using magnetic bead technology for the biological sample collection? That was the SAMI device, and in that case, they're using a traditional immunoassay. Now, on the SAMI device, I believe they procure their immunoassays from others. So that's one where I think it's Mountain Horse is the company you could look at to find out more about what technologies they are using inside of their immunoassay test strips. But in that case, it's usually just a traditional immunoassay. Hayden had another question, which was, I understand the majority of biological agents takes weeks or days to become obvious with signs or symptoms. Is there any that can show up almost immediately that you all know of? So reality is most biological agents that show up immediately are things like, there's Scott again. So things like toxins. So the toxins can show up immediately via inhalation, things of that nature. But in terms of traditional bacteria, viruses, and things, they generally have an incubation period in the body before the signs and symptoms show. There are a couple of things that, let's say, Salmonella, E. coli, those things may be hours, like 12-hour time periods, which would be less than days or weeks. But again, most of them, there's that incubation period in time the body that will be the issue. And so we don't know, if you just go back to the Salmonella events out west where they sprayed it on the food court, the salad bar, that's one of those things where you literally didn't know right away, and then you have to use epidemiology to follow back and figure out where each of these people ate that showed the use.
Yeah, Christina, I'm back. For some reason,
Okay.
I got dropped. Sorry. I don't know if you missed some of my previous response or not. I don't know when I
did. And so we were talking about the signal and about the fact that you were looking at the signal from more of a theoretical perspective, but not in that one slide. But that reality is there's a whole bunch of machine learning behind it, and that you're capturing all of that information.
Yeah, and I don't know if it came through or not, but it is possible for us to trigger even at what you normally will see in the environment is there'll be ebbs and flows, higher and lower levels, and so it's still possible for us to trigger when it's at a low level because of the algorithm and what it's looking for and how it works. So it's not really dependent upon it, but what we demonstrated there was just a hypothetical in PowerPoint, not necessarily an actual event in that case.
Okay, I think that was actually our last question for now. So with that, I just want to thank everybody for hanging out with us for the last hour and 15 minutes. And we will capture all of this on the video. It'll take a little bit of time for me to crunch that video down and to make it available to the ProEngine team for sharing. But we do have a variety of different things that we're working on, and all videos that we do for this training program are available and freely ready for all of you. So thank you all again, and stay safe out there, and happy holidays to all of you.